Okay, hi everybody. Let's pick up where we left off on our talk about medical terms used in cancer. So we were just talking about some statistics and also some key terms regarding cancer, and we left off talking about the age standardized incidence rates. And so again, looking at this image here, we can see that the population distribution across Canada varies greatly. We live in the most densely populated region in the country, but if we recognize that particular regions maybe have a larger elderly population than others, and then we perform age standardization, now we can see that incidence rates are actually more similar across different regions. Now, there will be some variation. For example, we can see here in Ontario, 545.9 per 100,000, or 91,600 new cases in the given year of 2021. And that was a little bit lower, for example, here, 482.8 per 100,000 in Saskatchewan. There could be many reasons for this. In terms of the environment, if you're in an area with higher pollutants, more factories, and so forth, things like that increase cancer risk. But when we standardize it by age, we see that the numbers are a lot more similar across different regions. And so age standardization helps us to create a more specific measure of the incidence of cancer in various regions. So now here we can also standardize the mortality rates for all cancers combined. And something I wanted to point out with this graph here is this shows in blue males, the orange represents both sexes, and then the green line here represents females. And we're looking at the rate per 100,000 individuals, and this is representing from the years 1994 to 2019. And the key takeaway here is that we can see that age standardized mortality rates are reducing, right? The mortality rate is reducing over time from 1984 to 2019. Now, certain types of cancer still remain to be very difficult to treat. And as we saw, for example, with pancreatic cancer, diagnosis is more largely or closely tied with mortality rates. So the incidence rate and the mortality rates were a lot more similar for certain types of cancers. But we can see for other types of cancer, such as here, female breast cancer with new treatments and advances in modern medicine, we can see that the net survival rates are higher. And survival duration over years is also higher for given types of cancer. Some, again, especially pancreatic cancer being more difficult to treat, the net survival rates are a lot lower. But we are seeing that the predicted net survival for the leading causes of cancer in Canada, depending on the type of cancer, things have become a little bit better with modern medicine. So something to consider as kind of a checkpoint question here. So a certain type of cancer has a low incidence, but high mortality. What might this suggest about early detection and treatment effectiveness? So perhaps consider this question, maybe pause the video, think about what we've talked about in the last video, and we'll come back and chat about it. Okay, so the question being, if a cancer has a low incidence but high mortality, what might this suggest about the early detection or about treatment effectiveness? So if we think about the new terms that we have gone over in this PowerPoint, and we think about how low incidence indicating that not many people are developing it, but high mortality suggesting that a large proportion of those who do develop this type of cancer will die from it. Taken together, this can perhaps present that the cancer is diagnosed late when it's already in the advanced or impossible to treat stage, or perhaps if there's no effective screening test or symptoms appear late, or the cancer progresses aggressively. So there are many different aspects here. But for example, we talked about with pancreatic cancer, right? How the mortality rate was quite high across for individuals who are diagnosed with pancreatic cancer. But with advancements such as methods of screening for and early detection, we can perhaps over time start to see those reduced mortality rates that we were seeing in some of those charts that we were looking at. And so those cancers with higher mortality tend to be associated with a little bit lower treatment effectiveness. But over time, we are seeing trends, especially for cancers like breast cancer, for example, where we're seeing that with modern medicine and modern screening techniques, we are seeing a lowering of those mortality rates. Okay, so let's dive into some of the basic biology of cancer cells and of cancer. So let's talk a little bit more and begin to understand what cancer actually is. So the biology behind it and what causes it. So cancer is essentially uncontrolled cell division. Here we are looking at skin cells. So these are, if you remember from your previous courses, these are stratified squamous epithelial cells that make up the epidermis of the skin. And these cells continuously divide. So they divide and then they push up to the surface. And then these cells at the top surface of the skin become flattened and actually die. And so these cells at the top here are filled with keratin protein. And this dead cell layer with keratin is what provides protection. So our skin is not raw. So that dead cell layer provides some like abrasion protection for our skin, if you will. So this is basically the cell turnover of the skin. Cells continuously divide, push up to the surface, become flattened, die off, and so forth. But let's go over and review the cell cycle. So somatic body cells divide through a process called mitosis, and cell division is a very controlled process. So once a cell gets the signal to divide, it will go through the G1 phase, or growth 1, S phase, G2, and then mitosis. And mitosis is where the division of the nucleus occurs. When the cytoplasm divides in cytokinesis, this is where we form two identical cells. But some cells in our body will remain in this G0. This little zero is, we call it naught, so G0 phase. And that is cell cycle arrest. So if the cell doesn't need to divide, it won't. And some cells won't, and they may die off eventually, or they stay in cell cycle arrest where they are not dividing and they're just quiescent. Now, as you would have learned, also when you learned mitosis previously, interphase has checkpoints along the way. So these checkpoints are going to search for any mistakes that have occurred along the way as the cell duplicates its contents, as the chromosomes are duplicated. There are many checkpoints that ensure that the cell has gone through these stages properly. If there are mistakes found along the way, the cell may undergo what's called apoptosis. Let me write that on the slide here. So apoptosis is the term for programmed cell death. So essentially, it should happen if there are mistakes in the DNA. Apoptosis. So if there are mistakes in the DNA, for example, if there are mistakes in our chromosomes, apoptosis is good. We don't want those cells that have errors in them to continue to divide and proliferate. If everything is okay and our cell went through interphase appropriately, then it will continue on and it can divide. And then one of the two cells can itself divide and so forth. But this process, and the key here to note is that this process is very controlled. There are checkpoints along the way, proteins that ensure that the cell is dividing properly, but also if it hasn't, or if a certain growth phase has an error that's occurred in it, or if S phase, duplication, occurred incorrectly, then that cell will undergo programmed cell death or apoptosis. So cell death is not necessarily a bad thing. It's a method of controlling cell division and ensuring that healthy cells move forward and cells that have any errors do not move forward in cell division and are killed. Programmed cell death. Now, cancer is uncontrolled cell division. So the cells begin to divide uncontrollably regardless of these checkpoints. They don't need signals to divide. The system is essentially hijacked and faulty, so the cell just divides no matter what, even if there are errors. And this is how cancer develops. If the cell cycle is not controlled, so the cells keep dividing, and as they continue to divide and there's no checkpoints or there's no signals, they're insensitive to signals telling them to stop dividing, they'll just continue dividing. They are not entering this quiescent cell cycle arrest phase. They just keep dividing. And eventually we form a mass or a tumor. If it gets big enough, or if some cells slip away, those cells can travel to a different region of the body and form a tumor there. So this is essentially the kind of hallmark of cancer, is this uncontrolled cell division. So some other hallmarks here. We have self-sufficiency. So cancer cells don't require growth signals. They just continue to move through the cell cycle. So they're insensitive to signals telling them to divide or to stop dividing. So they're insensitive to anti-growth cell signals as well that tell them to not divide. It's just continued division. Another feature here would be tissue invasion and metastasis. So this is the spreading of cancer cells. So cancer cells can migrate to other regions of the body and begin to form a growth or tumor in another region. Limitless replication. So it can continue on and on through the cell cycle. Again, insensitive to signals to stop. Sustained angiogenesis. So angio meaning blood vessel. Genesis means to create. So these tumors that are formed then develop blood vessels through the tissue, and these blood vessels will feed the tissue and maintain the nutrients and essentially the cancer growth. So giving that growth the nutrients that it needs. Again, metastasis, so breaking down the word metastasis, meta meaning change, stasis means state or transition. So essentially the movement and spreading of the cells to other locations of the body is what is meant by metastasis. So these are the common things that we see with cancer cells. They're not responsive to any anti-growth signals and just continue to divide, invading other tissues, and they also can sustain themselves. So sustained angiogenesis, gaining the nutrients that they need. And then this other one here, evading apoptosis, right? So they're not sensitive to signals telling the cell to die off. If there's errors, despite having errors, these cells continue to divide. Okay, so to understand cancer biology better, we need to understand how normal cells control cell division. And there are two major classes of genes that are responsible for controlling normal cell division. This includes tumor suppressor genes and proto-oncogenes. So let's start with tumor suppressor genes. Tumor suppressor genes are genes whose normal function is to inhibit cell division, so inhibit mitosis. In other words, think of them as brakes on the cell cycle. So our tumor suppressor genes code for proteins that slow or stop the cell cycle when conditions aren't right. So these genes hold a code for proteins which will stop the cell cycle when there are errors, for example, or DNA damage. So they're going to monitor the integrity of DNA, and they'll trigger DNA repair pathways when necessary. If the damage is too severe for repair, they may initiate apoptosis or programmed cell death. So if it's beyond repair, then the cell will be killed off. This is a protective mechanism. So if a cell has significant DNA damage, it should not divide and pass that damage on to daughter cells. Tumor suppressor genes also help ensure that cells only divide when they receive the correct signals. So they ensure that division happens in the correct tissue at the right time and only under very tightly controlled conditions. So not only do our tumor suppressor genes inhibit cell division and stop the cell cycle when things aren't right, but they also ensure that cells only divide when they receive the correct signals to divide in the right place at the right time. Now, a classic example of a tumor suppressor gene is P53. P53 is a gene that is going to largely help to regulate cell division, working to inhibit mitosis and restrict when and where cells can divide, really regulating cell division under tightly controlled conditions. Now, we also have proto-oncogenes. Proto means first, onco means cancer, plus gene. So proto-oncogenes sometimes get a bad reputation, but they are essential genes. So again, we're talking about in normal cell conditions. Proto-oncogenes are genes that promote cell division and survival. So these genes actually promote the division of cells, and they kind of act like a gas pedal on the cell cycle if the tumor suppressor genes were the brakes. So our proto-oncogenes code for proteins that are involved in growth factor signaling, cell cycle progression, so moving the cell cycle forward, cellular responses to signals that indicate for the cell to divide, so growth factors and things like this. So they, importantly though, proto-oncogenes do not, cause cancer or are associated with cancer when they're functioning normally, even though they are pro-cell division, if you will. They're highly regulated so that it's ensuring that they are only activated under very specific conditions, turned on temporarily and precisely when cell division is needed. For example, during growth, tissue repair, wound healing, so ensuring that cell division happens when it should happen. Problems could arise when these proto-oncogenes become overactive or permanently switched on. So issues can happen with either proto-oncogenes or tumor suppressor genes, but it's important to note that these genes are vital in normal cell activity. So tumor suppressor genes will inhibit mitosis and control to ensure that cell division is happening when it's needed and where it's needed. Proto-oncogenes work in opposition to tumor suppressor genes, and they promote cell division, but they're going to promote cell division where it's needed and where it's necessary, like to repair a wound or tissue repair. It's only when there are mutations in these genes that problems arise, and which is where cancer perhaps may develop. So we have a fine balance of these genes to determine if a normal cell divides. But if a gene becomes mutated, then the DNA changes, and that change to the gene product is then, produced, translated. That gene product, which is the protein, if it's mutated, things can begin to go wrong. So in cancer, tumor suppressor genes are mutated in certain types of cancer. So the DNA changes, and it could be one small base pair change in the DNA, specifically in a tumor suppressor gene. When it's mutated, this leads to dysfunction, and we're no longer able to stop the cell from dividing. The mutated gene does not behave the way it should. The gene product does not perform its normal role. And in this case, the suppression of the cell cycle or controlling cell division now goes haywire. So now the cell's dividing without anything keeping it from dividing. So essentially, mutations in tumor suppressor genes means that we are not suppressing the cell cycle as we should be. We are not suppressing cell division as we should be, or regulating it and controlling it as we should be. So now the cell will divide uncontrollably. With proto-oncogenes, in a cancerous state, these genes can become mutated and dysfunctional, and in this case, they tend to remain constantly on. So now instead of controlling going through the cell cycle, it is now always promoting cell division. So mutated proto-oncogenes, they tend to remain constantly turned on, always promoting cell division. And so in this case, the gene is then referred to as an oncogene. This is an oncogene causes cancer. We take the proto off of it. And so we can see how under normal conditions, these genes are very helpful and aid in the control of the cell cycle. But when mutations arise in these genes, this is what is associated with a cancerous state. So that altered balance between tumor suppressors and proto-oncogenes caused by mutations in the DNA drive cancer. So normally things kind of look like this, right? But if it's mutated, there's no more control. So typically our tumor suppressor genes will inhibit cell division, and our proto-oncogenes stimulate cell division. But if our tumor suppressors become mutated, then you end up with, we don't have any inhibition really of cell division from our tumor suppressors. And therefore, when we don't have that inhibition, cell division is uncontrolled. On the other end, if there is a mutation in our proto-oncogenes, they become permanently turned on. And so we get overstimulation of cell division. So these genes have actually been identified in more than 50% of cancer cells, specifically mutations to p53, that tumor suppressor gene that we talked about, is highly associated with cancer. So when cell damage occurs in a normal cell, so p53 being a tumor suppressor gene, normally p53 will halt the cell cycle so that the damage can be repaired in one of the cell cycle checkpoints. This is in a normal cell. If the damage can't be repaired, p53 will initiate apoptosis when the cell cannot be repaired. And in this case, apoptosis is a good thing. We don't want a damaged cell to continue to divide. But if p53 becomes mutated, the mutated form does not arrest the cell cycle. The cell cycle continues and cells can become cancerous if they continue to divide, producing more damaged cells and so forth. So mutated p53 no longer suppresses the cell cycle. So this is a nice illustration that kind of shows that interaction between the tumor suppressors and the proto-oncogenes. So in normal genes, again, the tumor suppressors are going to suppress cell division, control cell division only when it's required. So think of it like the brakes of a car. In a healthy tissue, the brake is working, and so we're halting cell division and keeping it at a pace that is required and what we need. The proto-oncogenes act as an accelerator of cell division. If we need to heal a tissue, if we have a wound, for growth, we need cell division. And so our proto-oncogenes will help with that and accelerate cell division, but they're still tightly controlled. Remember, the key here is control, and they're mostly turned off. In a mutated state, now imagine our tumor suppressor gene is as if the brake isn't working, so we no longer have control on the brakes. So now we're no longer suppressing cell division, and cell division becomes uncontrollable. And at the same time, the accelerator is jammed, so we're constantly pushing on the gas pedal and therefore further stimulating cell division to occur through mutations in our oncogenes, or forming an oncogene. So a little activity that I have for you guys, and a little checkpoint question: genes associated with cancer. So I recommend filling out this table on your own, either in your notebook or, just jotting down the normal role of each of these genes and also the cancer-causing mutation. So recognizing that these genes are vital for normal cell functioning, but if mutations arise in either of these two genes, this is where a cancerous state may develop. Okay. So here we're looking at normal homeostasis of cells. Homeostasis is referring to maintaining an internal balance around a certain set point in the body. So normal cells go through this process of cell homeostasis with cell death, normal cell division. But in normal cell division, we have some cells, as we've said, that undergo normal cell death. Maybe chromosomes didn't divide properly, errors occur during the cell cycle, and so it's normal for apoptosis to occur. And so we have this balance between normal cell division and apoptosis, and we maintain a healthy, say, number of cells and a healthy population of cells. In tumor formation, you have essentially two different scenarios that can happen, one of which that disrupts cell homeostasis is increased cell division. But apoptosis might still be occurring at a normal rate. So in this case, say if we have a mutation in our proto-oncogene, we're constantly stimulating cell division. Apoptosis is still happening normally, but we still end up with tumor formation because we have that driver of that increased cell division. The rate of apoptosis can't keep up with the rate of increased cell division. Second scenario may be that there's a mutation causing decreased apoptosis. So now cells that are damaged are not properly undergoing programmed cell death, and so those cells are dividing, and we still have normal rates of cell division, but we're not, cells are not undergoing apoptosis that should be, and so the overall net number of cells, again, is greater, and this results in tumor as well. So big hallmark, your big takeaway is that cancer cells are not performing the normal functions of the tissue that they are in, and we see this disruption of cell homeostasis. So now coming back to this photo, hopefully this makes a bit more sense now. Here, the abnormal cell division or significant decrease in apoptosis either can cause cancer. And the big thing and the big takeaway here is that it's about a lack of regulation and control of cell division. So this slide is talking about how cancer is a progression. Now, something to note is that there are phases that researchers and scientists have discovered with cancer cells. And we'll go through kind of what each of these phases is here. So the first is hyperplasia here. So in the hyperplasia state, hyper meaning more, plasia meaning growth. This is described as an increase in cell numbers and growth compared to normal tissue. So here we have normal tissue on the left, and here we have tissue in hyperplasia. The cell structure and tissue structure looks largely unchanged, but there is some increase in cell number. So this won't be that remarkable where it won't be that visible of a change, but there is more growth beginning. So this is kind of a very, very early stage that we're looking at. Next, we have dysplasia. Dys, as we've seen as a prefix, means disordered. So now we have a continuation of hyperplasia with some disordered growth. We see buildups, right? It's very disorderly buildups of extra cells. Cells now look even different. Overall, as a tissue, this looks a lot different. So at both the single cell level and the tissue level, we're seeing a lot of disorder. And finally, in a carcinoma tissue or carcinoma in situ, carcinoma means cancerous tumor, right? Oma as a suffix means tumor. Carcin referring to cancer, so cancerous tumor. In situ means in place. So in situ means in place. And carcinoma in situ is a continuation of previous dysplasia. And depending on the tissue, this may be referred to as stage 0 cancer. So it's not quite yet fully cancer as it's contained within the tissue and does not spread, but can become cancerous over time. So this is where we begin to categorize the tissue as stage 0. It's contained, it doesn't spread, but it can become metastatic and spread over time. So here's another photo showing this. We move to hyperplasia, where the cells divide more rapidly, and then to dysplasia, where we see a lot more disorder, and cells begin to change form, and it's this disordered growth stage. And then if the in situ cancer, where the cells stay in one place but are largely, there's a lot of overgrowth, very disordered tissue. If this progresses, then it can become malignant. And a malignant tumor containing these malignant cancer cells, if those cells invade other tissues, reach a blood vessel like here, now they can travel to other parts of the body through metastasis. So form and travel to different sites in the body. Okay, so I'm going to pause this video here now that we've discussed a lot of the basis of the cancer biology, the different genes that are involved in cancer. In this next video, part 3, we're going to talk about cancer terms and also some screening and technologies that we use for cancer diagnosis.